The overall goal of this procedure is to treat amphibians infected with the amphibian chytrid fungus Batrachochytrium dendrobatidis, or BD, using the antifungal chemotherapy Itraconazole. This is accomplished by first growing BD cultures and inoculating animals with the fungal pathogen. The second step is to test animals using DNA extraction and qPCR on skin swabs to ensure BD infection. Next, the infected animals are treated by bathing the animals with 0.0025% itraconazole solution for 5 minutes a day for 6 days. The final step is to monitor the infection status of the treated animals by performing qPCR analysis on skin swabs for a minimum of 4 weeks after the completion of treatment to ensure successful treatment of the animal. This procedure demonstrates that a reduced concentration and shorter treatment time of the antifungal itraconazole is efficient for the treatment of animals infected with BD.
The main advantage of using this treatment regimen over the recommended treatment dose, which is a 0.01% itraconazole solution for 11 days, is that a reduced concentration of 1/4 that recommended dose and a reduced treatment time to 6 days is still effective at curing BD infection, but also minimizes the negative side effects of using the recommended treatment dose.
After incubating the BD in tryptone broth as outlined in the text protocol and checking for zoospore movement, flood each plate with 5 milliliters of aged tap water and wait for 10 minutes to allow zoospores to enter the media. Next, pour the zoospore suspension into a portable container.